last time with the sarcomere. Okay. Where is my thing? Okay. So muscle has to be integrated with nervous system activity because, we only want muscles to contract when there's a reason for them to do so. So all skeletal muscle is voluntary. In other words, it does not contract on its own. Unlike cardiac muscle and unlike smooth muscle, which do contract on their own, skeletal muscle requires nervous system input to begin contraction. And the connection between the nervous system and the muscular system takes place at what we call the neuromuscular junction, which is also called the motor end plate. So those things are all the same. The neuromuscular junction is where the nervous system, particularly the lower motor neurons that are found in the spinal cord, their axons come very, very close to the muscle at the neuromuscular junction or the motor end plate. So those of you that have had some, biology in the past, which is probably all of you, you'll remember that neurons connect to other neurons at a gap called a synapse. Well, at the neuromuscular junction, we have a synapse, but instead of being neuron to neuron, it's neuron to muscle cell. But you'll see here in a little while how the two are very, very similar. So essentially, the neuromuscular junction is a synapse of nervous system tissue onto skeletal muscle tissue. So if we zoom in and look at that, what we can see is, well, here, let's zoom out. So here's a motor neuron, cell body is in the spinal cord, long axon going through a peripheral nerve, and then at a part of this muscle fiber, and remember a muscle fiber is a muscle cell, we have this tree-like expansion of the axon. And then if we zoom in to one of these expansions, we can see that here we have a branch of that axon. We have this gap, and then on the muscle, we have a region called the motor end plate, which is going to receive the signal, so to speak, from the neuron. All right. So if we zoom in and we look at the events that take place at that synapse or that neuromuscular junction, we can see a process that is very similar to what we'll talk about in nervous system synapses here in a few weeks. So what we're looking at, here is the muscle cell down here. This is the nerve cell, so a branch of that axon that's coming very, very close to the muscle. Inside that axon, we have some vesicles that contain acetylcholine. Acetylcholine is a neurotransmitter. It's used exclusively, it's the only neurotransmitter that's used in the skeletal muscle system. So at the neuromuscular junction, the only neurotransmitter that you ever find there is acetylcholine. When we get into our nervous system chapters in the next units, we'll see that there are lots of different kinds of neurotransmitters that do lots of different kinds of things. But at the neuromuscular junction, it's always acetylcholine that is the neurotransmitter. So we see that there in red. Now on the muscle cell membrane over here, we have a number of receptors to acetylcholine. So we can see these are little spheres, and these receptors kind of have a rounded place at the top. That's to show you that the two are going to fit together. So when acetylcholine reaches these receptors, it's going to trigger something, it's going to make something happen. All right. And then the last layer are these little moon-shaped molecules. This is acetylcholinesterase. Acetylcholinesterase, which we abbreviate ACHE, it breaks down acetylcholine. So when acetylcholine joins up with this little crescent shape, that acetylcholine is going to be broken. Once it's broken, it's inactivated at that point. So what we'll see here in a minute is it helps to turn off the signal for contraction. All right, so this is how things are at rest. So the nervous system is not stimulating this muscle to do anything. All right. Once the signal from the brain comes down that motor neuron, eventually it's going to reach the end point here of the branch of that axon. So what we see here in red is what we call an action potential. Now we're going to talk at length about action potentials, what that is, how that works, and what that means when we get into our nervous system physiology section. But for now, just think of an action potential is a change in the cell membrane that can trigger other changes. Specifically, it's a change in the cell membrane's voltage. Because of the way the ions are distributed in our cells, across the cell membrane, there's always a small electrical difference or voltage. So an action potential is just a change in the voltage of the membrane. That change can make all different kinds of things happen, as we'll see here in a minute. So the brain has instructed a muscle to fire. So that change in membrane potential or change in membrane voltage travels down the axon of the motor neuron and then reaches this branching area called the motor end plate. Once that signal arrives, it triggers the release of acetylcholine at this neuromuscular junction or at this synapse. So this arriving signal from the brain is what triggers these vesicles to join up with the cell membrane and actually dump their contents into the synaptic cleft or the space between the nerve cell and the muscle cell. So we see the acetylcholine molecules being emptied into this space. So they travel across this space by diffusion, and the acetylcholine molecules then link up with those receptors that we see here. Once that occurs, the cell membrane now of the muscle cell has a change in voltage, so a change in membrane potential. And that occurs because as acetylcholine joins up with these receptors, sodium channels are open, so sodium basically comes rushing into the cell and causes a change in that voltage. So just like we saw this membrane potential change coming down the axon of the neuron, now we're seeing a membrane potential change begin on the muscle cell as opposed to the nerve cell. All right, so that membrane potential change begins. You can see they've colored the membrane red to kind of show you that. And it doesn't stay put. This membrane voltage change moves. So first it occurs here, but then what we see is that action potential then spreads along the whole cell membrane. So it starts right here at this point, but then that action potential spreads all the way down the length of that muscle fiber. So that signal traverses that whole muscle fiber. Now, at that same time, while this action potential is propagating or moving down the cell membrane, we can see that now the acetylcholinesterase has joined up with the acetylcholine and is breaking it down. So once the signal is received here and the molecule is released, the neurotransmitter released, the next phase is to now turn that signal back off. And that occurs in two ways. One is the acetylcholine that is in the cleft here, or the gap, is reabsorbed. So that it ends up back in the axon where it started. And then some of the acetylcholine is actually broken down into its component parts, and then those component parts are reabsorbed so that they can be put back together and be ready to use again. So we have a motor neuron fires, that action potential travels down that axon. Eventually it triggers action potential on the cell membrane of the muscle, and then that traverses the whole muscle cell. So what's the point of all that? You know, how does that then trigger muscle contraction? Well, it's in this last part here that it makes sense. This action potential that's traveling down the membrane is what triggers calcium to be released from the sarcoplasmic reticulum that we talked about last time. So the action potential from the nervous system becomes an action potential on the muscle cell, And then that action potential on the muscle cell is what triggers calcium to be released around those sarcomeres. So if we kind of put that together in a movie, we can see it a little bit clearer, I think. All right.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3.
That step isn't in your book, but we'll pick it up when we get to the nervous system. So the trigger for acetylcholine release is actually calcium channels. That action potential comes down from the brain, and when it reaches the end of that axon, it opens calcium channels. Calcium comes in, and that's what triggers the acetylcholine to be released. So, this didn't come from the same publisher as your book, so the steps are a little different. But
calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholine esterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
all right, so that's events at the neuromuscular junction. So the end result of all that is that is that an action potential is now spreading across the sarcolemma, or the cell membrane of the muscle cell. And that's what we're going to see triggering the next step in this contraction cycle of releasing calcium so that the muscle can actually contract. All right, let's do a first question here. Troponin has which of those things? Which of those things are true about troponin? All right, I'm going to kind of skip this one because we haven't actually talked about this yet. So sorry. The correct answer there is D. Troponin has a calcium binding site, but we haven't talked about that yet. So bad question. Sorry about that. All right, this is one's better. Which of the following is composed of myosin molecules? Jump in there, last few people, if you're not in already. All right, so myosin makes up the thick filaments. The thin filaments have multiple parts. I mean, most of the thin filament is actin, but there's also tropomyosin and there's troponin, all on the thin filament side of things. On the thick filament side of things, all it is myosin. All right. Here's a good one from the sarcomere. The region of the sarcomere that contains both actin and myosin myofilaments is called which of those things? Great, 10 seconds. All right, good. Most of you got that. The correct answer there is B, the A band. So the A band is all of the thick filaments, and in the edge regions of the A band, you have both thin and thick filaments are all in there. The I band is only thin filaments. So under the microscope, the A band looks dark and the I band looks light. So it gives skeletal muscle that alternating pattern of light and dark. The Z disc or Z line is what connects the thin filaments, and the M line is what connects the thick filaments. And then the H zone is in the middle of the A band where there are only thick filaments, but we didn't talk very much about that. All right. So excitation, contraction, coupling. So far, we've seen this muscle cell get excited. You know, it was excited by the acetylcholine that was released in response to that action potential traveling down that motor neuron. So the cell membrane has now been excited. Well, what we want to have is we want to have that muscle contract. So the way we link excitation with contraction is through the T tubules and the sarcoplasmic reticulum. All right, so here's a myofibril, here's another. And we talked last time about how this blue system of membranes is called the sarcoplasmic reticulum. You know, it has an inside, and inside here, there's a ton of calcium that's been sequestered, you know, hidden from the myofibrils. And then dipping down from the cell membrane, we have these extensions of the cell membrane that are called T tubules. So we have this pattern like this. So as that excitation from the motor end plate spreads across the cell membrane, it falls down into these T tubules because the T tubules are technically cell membrane too. So that signal dips down into the muscle fibers, and then it comes very, very close then to the sarcoplasmic reticulum, which is, you know, we see here and here. So in response to that excitation that's traveled down the T tubules, the sarcoplasmic reticulum responds to that it's because it's right next to it, and it responds to that change by releasing calcium. So when the action potential reaches the T tubules near the sarcoplasmic reticulum, calcium channels open, and because there's much more calcium inside the SR than there is outside, calcium goes rushing out. And then that calcium is then what triggers the actual contraction. So just looking at that excitation-contraction coupling in fancy graphics, watch this little movie.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as the sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
All right, so nervous system activity creates muscle cell membrane activity, which then in turn releases calcium into the sarcoplasmic reticulum. So why does that matter? Well, it's in the presence of calcium, contraction can actually occur. All right, so we're going to look at how does that happen. First up is in the resting muscle. So what we have zoomed in on here is the thick, thin filament space. All right, so back up our. So we have a sarcomere, a simple one, looks like that. So that's thick filaments in black. Thin filaments are in red, right? Connected by the Z line out here, M line. So in our picture here, we have zoomed in to this spot right here. So, well, actually here, it's more like that. So we're looking at the spot where the thin filaments, which we see in the middle of our pictures, are interacting with the thick filaments that we see at the top and bottom. So myosin is in purple here, and actin is in red and yellow, and tropomyosin is in this kind of green here. All right, so here's a thick filament with its myosin head sticking out, and then our thin filament in the middle. So most of that thin filament is actin. So it's this chain of pearls, so to speak. And each one of those pearls has an active site. So when the muscle is at rest, the setup is like this. So nothing is happening because all of the active sites are being blocked. So the two molecules that want to interact here are the myosin head and the active site on actin. Those are the two pieces that want to get together to make contraction happen. But as long as there's no calcium around, troponin keeps tropomyosin locked in its covering position. We call it the covering position because it's covering those active sites so that myosin and actin cannot interact. But now, after excitation, contraction, coupling, in other words, that excitation of the cell membrane has caused calcium to be released. Well, when calcium is present, which we see here in green, calcium links up with troponin, and when it does, it causes, or troponin causes tropomyosin to kind of slip into the groove of actin. You know, if you think of a twisted string of pearls, there's a kind of groove that you can follow around. Well, when calcium is present, tropomyosin slips into that groove, and now we see lots of yellow sites on actin. So in the presence of calcium, the active sites are exposed as troponin and tropomyosin move, and then once those active sites are exposed, now contraction can happen. Because once exposed, the next step is myosin and actin connect to one another. So they want to all the time. You know, the chemistry there, if it wasn't for tropomyosin, the muscle would be contracted all the time. So in the presence of calcium, when those active sites are exposed, step one is what we call cross-bridge formation. And this is where the myosin head is attached to the active site on actin, or the myosin binding site on actin. Now, you'll see inside the myosin heads here, ADP plus P. We know that muscle contraction is going to require energy, and lots of it. And the energy molecule for the cell is ATP. So, and we'll see how that ATP gets used. But for now, the myosin head is holding on to an ADP and a P. They're part of that molecule. So once that connection occurs, now we have a connection between the thin filaments and the thick filaments. Okay, that's step one, the muscle, but we haven't contracted yet because nothing has moved. The actual movement occurs in the next phase. So after cross-bridge formation, the next thing that happens is that ADP and P are released from the myosin molecule. And when that occurs, the myosin head changes shape. It goes from an obtuse angle to an acute angle. So when the molecule is like this, it has an ADP plus P kind of hanging on it, right? Well, when this ADP and P are released, the myosin head changes into that shape, and then the ADP plus P are released, so they're let go. And this is how muscle actually shortens. This change in shape, albeit a subtle one, you know, from an obtuse angle to an acute angle, is what creates the pulling force that pulls the thin filaments are pulled in towards the center of the thick filaments, or the M line. All right. So the myosin heads pivot. This is also called the power stroke because this is where force is actually created. Now, of course, just one little ratcheting motion of a little tiny molecule is not going to do us very much good. You know, it's not going to do much. So we need to get this cycle repeating so that the actin filaments will continue to get pulled in towards the thick filaments and towards that M line in the middle of the sarcomere. So after the ADP plus P are released and the myosin head pivots, now the myosin head lets go. And when it lets go, it's now possible for it to reset or go back to its original shape. And the way that occurs is ATP joins up with this myosin head, and that's what triggers the myosin head to let go of the actin. That ATP is broken down but not released, so it's broken down into ADP plus P, and the energy that's released from that breakdown is stored as the myosin head in that cocked position. So when that ATP is broken down, instead of that energy just being released, it becomes a change in this molecule, this myosin molecule's shape. the head rotates back to its resting position. And as long as calcium is around, we go immediately back to this one. You know, there could be an arrow right here. So as long as calcium is around, we go active site exposure, cross-bridge formation, myosin head pivoting, cross-bridge detachment, myosin reactivation by that ADP gets broken or ATP gets broken down, and then we go right back to this active site exposure again. So this process repeats. The myosin head grabs hold again, moves, it shifts its shape, pulls on that actin thread, lets go, and then round and round and round. So this is essentially muscle contraction, or the cross-bridge cycle. And this whole view of muscle is called the sliding filament theory, which is not a theory anymore because we've actually seen it happen. But so this is cross-bridge cycle. We just talked about.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick
Okay, so thick filaments in the center. These are the thin filaments off to the side. So this is the Z line. The M line would be right here in the middle, just to orient you because the picture looks a little different than what's in your book. Okay.
Myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions released from the sarcoplasmic reticulum bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
All right, so that's the cross-bridge cycle, or what actually causes the thin filaments to get pulled in towards each other and towards the thick filaments, are all those little myosin heads ratcheting and ratcheting and ratcheting, and sort of one at a time pulling those thin filaments in. And because there are so many myosin heads and there are so many sarcomeres, this even though we zoom in and we look at just one molecule at a time, but there are millions, hundreds of millions of even these myosin heads interacting all the time, creating that what we consider muscle contraction. All right, so that's the scientific version, and I have to share this one. This one is a little bit more fun, but it's a little silly too. Okay. At least I think it's funny. So this is muscle physiology in a nutshell.
Action potential rise with the charge and it's upset. It's going off about a signal that the brain sent, and into the extracellular fluid goes Ca2. Influx of calcium cause vesicles to leak ACH into the synaptic cleft and to the motor end plate it gets bound to. Then there are receptors, gated ion channels. They open and let ions flow into the cytosol, flowing all around until the cell begins to find that this electrical difference means it's devoltile. Electricity begins to spread to T-tubule, releasing calcium from the SRKGC. It's so exciting. It's so exciting.
The calcium is Troponin C on the filaments of the myofibrils, and in an allosteric unblocks the binding sites. It's so easy. Myosin unblocked, it's definitely about to go down. Happens all the time when ADP is bound to open sites on the filament set. ATP makes myosin stroke.
Release of ADP and inorganic phosphate. ATP energy helps to move the head back. Myosin releases from actin all the time. Now let's do it again and let myosin and find it. You see that it happens all the time. Happens all the time. Now can't you see? It's so easy.
It's easy, right?
Made my day.
All right, so that's the more fun version. Okay. All right, so let's do some questions about that stuff. All right, the active sites to which cross bridges attach are found on which of those things? Did you notice in that video that the guy was totally the weakest link?
Wow.
All right, jump in there if you haven't. All right, so the cross bridge is the connection between the myosin head and the actin filament. So the active site that makes that happen is found on B. The actin myofilaments have a myosin binding site. And that myosin binding site in the relaxed muscle is covered by tropomyosin. But in the presence of calcium, that binding site is exposed, and it starts that cross-bridge cycle, which actually shortens the myofibril. Oh boy, here's a big one. All right, so put these events in the correct order. So you have to read and think on this one. I'll give you a little more time on this one since it involves some reading. All right, jump in there, last. All right. So there are a couple of spots in this first semester where we have these long strings of cause and effect. You know, one thing causes another, which causes another, which causes another, and so on and so on. And this muscle physiology stuff is very much like that. You know, the lower motor neuron starts the process. You know, that action potential arrives at the neuromuscular junction, you know, and that triggers an excitation of the skeletal muscle membrane. That excitation travels all around the skeletal muscle fiber, down into those T tubules, comes right next to the sarcoplasmic reticulum, and that the SR becomes more permeable to calcium, so calcium is released. That calcium diffuses around the myofibril, interacts or attaches to troponin. Troponin moves tropomyosin, exposing those myosin binding sites, and that starts the cross-bridge cycle. So the correct answer here is C. So step one is the action potential comes across the membrane down into the T tubule. That causes, or did I say C? Sorry, it should be B. Because then the next thing is that the sarcoplasmic reticulum becomes more permeable. Number 4, calcium ions diffuse. Those ions bind to troponin, that's number 2. And then the troponin-tropomyosin complex moves. So the correct answer there is B. All right. Complicated question, I know. Okay. So now we know a lot. You know, we've now seen how a signal from a neuron in the brain can actually create a change in the length of a muscle by triggering that cross-bridge cycle through that excitation-contraction coupling. So neural control causes a release of calcium, and that calcium triggers a physical change to the muscle that actually causes that shortening. So then now we're going to zoom out, and we're going to look at now how does the muscle as an organ function? Because so far what we've been looking at is a myofibril. In other words, one contracting thread in a skeletal muscle fiber. So one tiny little piece of the muscle. Now it's important because the way that one tiny little piece works is the way all the other pieces in the muscle work too. But now that we've seen that system, we can take a step back and we can look at, now how does the muscle as an organ function? All right. So we've seen that calcium acts as a kind of switch. You know, when calcium is present, that muscle is going to be contracting. When calcium is absent, that muscle is going to be relaxed. But obviously there has to be more to it than that, because our muscles are not just on or off. You know, we can lift a paperclip or we can lift a 50-pound bag of sand. So clearly there's more to it than contracting or not contracting. And there are 3 main things that the body controls or that matter to how much force or contraction strength is actually generated. So one of those is muscle length, and then the others are the frequency of stimulation. In other words, like at that neuromuscular junction, the more rapidly the nervous system stimulates a muscle, the more it will contract. In other words, it will contract with greater force and greater speed the faster it's stimulated. And then inside of an individual muscle itself, not a muscle fiber, but a muscle that has many fibers, the brain can control how many fibers are actively involved as well. So all three things can be controlled, and we'll look at each one of those separately. We'll do length today, and then we'll finish up the rest next time. All right, because of the anatomy of the sarcomere, length matters. And it's easiest to see this at the two extremes. So if we took a muscle and we sort of crunched it and we made it as small as we could make it in its contracting dimension, we squeezed on the ends. Essentially what we would do is we would push all the pieces of the sarcomere together. So like what we see down here, remember this is normal. So thick filaments, thin filaments, and then some overlap space here where the actual contraction occurs. But if we take this sarcomere and squish it, what we end up with is the thin filaments and thick filaments are all pushed together. Now when a muscle is in this state, it can't contract because it can't get any smaller. All the thick filaments could pull on the thin filaments, but they're already as compressed as they can get. So a muscle that's in this state can no longer contract, so it can't generate any tension. Tension is just the force of contraction. As we get a little longer here, okay, now the thick filaments and thin filaments can interact, but even still, the thin filaments are already butting up against that M line, so they can't, be pulled on anymore. But then when we get up into normal, we have room to contract because the thin filaments and thick filaments have some space between them, and we have a way to contract. The myosin heads can get at these thin filaments. As we get a little longer, that starts to break down. As the thin filaments start to get pulled away from the thick filaments, now there is less zone of overlap. In other words, there's less space where the myosin heads and the actin can actually interact. So the muscle is going to get weaker because there's not as many pulling parts. And then if we continue to stretch that muscle, we'll actually separate the thin filaments from the thick filaments, and now nothing can happen. Now, anatomically, the body keeps muscles in this range right here, in the normal range. In other words, you can't compress your bicep like this, and you can't stretch it out like this either, at least not without doing some cutting of tendons and things like that. So the body keeps it in a normal range, but at the ends, there is still an effect here. So in other words, when a muscle gets too short, its ability to create force drops off, and when it gets too long, its ability to create force drops off. Now, we account for this in our actions and motions all the time. If you've ever gone to lift something heavy and you repositioned your body so you'd have better leverage, is oftentimes how people think. Some of what you're doing is you're keeping those muscles at that optimal length for generating force. All right, but we'll pick up on this next time, and we'll go from there.
Dr. Keith?
Yes?
This picture over here, that's what's when you're going like this, right? Is that constructed?
Well, it's actually, you can't get that bad without cutting something. But if I take your muscle and I squeeze it together, that's essentially what gets on the end.
So if you're going like this.
Oh, remember, if you think you might have left your bone sheet, folks, somebody did. If you figure out it was you, just email me.
So it's like on this side of the range, so that's
Yes, exactly. That muscle is very, very short, and you start to lose the ability to make as much strength that way.
You today. Thank you. And my year review at work, so I'm freaking out. Oh, I'm sure it'll be fine. And I had my stats exam this morning. Oh, how'd that go? I failed it. I know, I would have taken the class online. With the 34